Thanks everyone for participating, and thanks to Dr. Mikael Ström from Capitainer for today's presentation. I'm Ilaria Josue, and I'm a sales product and specialist for Prodotti Gianni. Prodotti Gianni has been working in the life science field for several years, and is the unique Italian distributor of Capitainer. Today's webinar is aimed at the presentation of the quantitative devices for the collection of dried blood samples developed by Capitainer. At the end of presentation, feel free to ask or put in chat box your questions, and Mikael will be happy to answer. For further information, you can contact me at the contact details that I will leave you in chat box. Okay. Thanks again for participating, and Mikael, if you are ready, you can start.
Yes, I am. Thank you for getting the chance to present here today about our self-sampling platform at Capitainer. Just a brief background, Capitainer is a company founded in the northern part of Europe, in Sweden, outside Stockholm. We stem from the beginning from a joint research project between two universities in the Stockholm area, the Royal Institute of Technology and the Karolinska Institute here in Stockholm. And the idea really came up when a professor working at the hospital laboratory, Professor Olof Beck, at the hospital on his way to the laboratory, passed the centrals for delivering a blood sample and saw the amount of people just sitting there and waiting to have a blood draw, and realized there must be a better way of providing your medical doctor with a sample. So to summarize why Capitainer was developed and the problem we try to give a better solution to, it's with traditional blood sampling, you are dependent on that the patient is traveling to a phlebotomy center, often located at the hospital, for a venous blood draw. The blood draw requires professional resources by the healthcare. You need to have a trained nurse or medical doctor to do a venous blood draw. The blood then needs to be shipped to the laboratory for analysis, and since wet blood is dependent on cold chain transportations and is not stable for so long time, it needs to be shipped rather fast to the laboratory for analysis. Capitainer is targeting the market where blood analysis does not need to be done immediately. So we are not targeting the emergency rooms, but anything within clinical monitoring, research studies, or consumer testing, where you need to have a monitoring of levels in your blood, but you're not dependent on having the result within hours. So how does it work? Giving a sample with Capitainer is easy. You get a sample pack sent home. You open the Capitainer card. It's based on finger pricking and finger blood. So you prick your finger with a lancet, which is included in the test kit. You apply a drop of blood to the card. The product always gives a confirmation of successful sampling, which is an extremely important feature in home sampling. It should be easy for the user to know that, yes, I succeeded with my sampling, or no, I need to contact the physician, study leader, etc., to get a new product. And then it's shipped by regular mail to the laboratory for analysis. I will share a video here. It's only a 1.5 minute video showing how you sample with Capitainer. So some of you might recognize quite a lot of the instructions in this video. And as you might then see, Capitainer is really based on the standard procedures of capillary sampling and of dried blood spot sampling. Dried blood spot sampling was introduced broadly in 1962 by Robert Guthrie for PKU testing of newborns. And this is the so-called heel prick that has been done in most countries since the 1960s in all newborns, where you screen for inherited errors of metabolism. In this case, it's screening for lacking an enzyme, which means that you cannot break down the amino acid phenylalanine. The PKU screening really revolutionized the development for these babies, that before you had the screening, they developed severe deficiencies, but with the screening and identification of this enzyme deficit, they could go on a low protein diet and develop and have a fully normal life, which really made a huge sense to thousands of babies around the world since then. However, standard dried blood spots are associated with some limitations. And the main limitation is that you don't know how much blood you have. On a standard dried blood spot card, you apply blood to the filter paper, and then it comes into the lab and you do a sub-punch from this. So you punch out a certain area of the dried blood spot and then assume that this will contain a certain amount of blood. This is only true to a certain extent. There are many factors that will affect how much blood that is located in this dried blood spot. And the main one is the hematocrit of the blood, where a blood with higher hematocrit is thicker and will spread less in the paper, while a blood with lower hematocrit is thinner and will spread more, then affecting how much blood that is located in the area that you punch out. There are also many possible sampling errors you can do, and sampling with standard dried blood spot cards requires quite a lot of knowledge by the user. And this means that standard dried blood spot analysis is only semi-quantitative in its nature. And I just also wanted to bring up regarding sampling errors. This is actually from a large Canadian COVID study that was done during the pandemic with standard dried blood spots. And this was the instruction manual sent home to the users just to show all the possible errors you could do when you did dried blood spot sampling, clearly showing that it's not easy in a user-friendly way of doing blood sampling. So also regarding analytical quality, we know a lot of people working in the lab and in the laboratory are very focused on the analytical precision, the precision of the method. But what we know is that the result quality will never be better than its sample. So if you have a poor and inaccurate sample, the result will still not be good even though the analytical lab work is done with high accuracy and precision. Only by combining a good analytical method and a good sample quality, you will receive high quality of the results. And Capitainer's idea and what we work towards is that a sample taken at home should always provide the same quality as a sample taken in the hospital. We have therefore developed what we called QDBS technology, Quantitative Dried Blood Spot Technologies. And it's really developed for accurate self-sampling. It's based on a microfluidic solution where a microcapillary meters an exact volume of blood and deliver this blood volume to a pre-cut dried blood spot disc, and you use this full metered sample volume for your analysis. The animated file here to the right shows how this works in our 10 microliter card, the function of that. So the idea is that you apply a drop of blood to the inlet. It will fill the metering channel. And on top of each membrane here, you have an excess blood membrane to the left and the QDBS sample disc to the right. What happens first after the blood is applied is that the membrane on top of the excess blood disc bursts, emptying the excess blood. Thereafter, the membrane on top of the sample disc bursts, then emptying the exact metered volume of blood to the sample disc. And this way at home, we can ensure that you always get a perfect volume of blood sampled at home. Even if your blood drop is 20 microliters, 30, 40, or 50 microliters, in this version of the card, 10 microliters will always end up in the sample disc. And if you add too little blood, the microcapillary is never filled and it's never emptied, so you will have no sample, never an underfilled sample. Compared to a standard dried blood spot card, the QDBS technology always also provides sample protection, so you cannot touch the sample as you can do with a standard dried blood spot card, and it's not exposed to the surfaces where you put the card, etc. Capitainer has so far developed 3 different products that are available on the market. The first one, Capitainer B, was released 2020 in February, just 1 month before the pandemic hit the world. We thereafter, approximately a year after, released Capitainer B Vanadate, which is the first product where we have incorporated a stabilizer in the card. So this is specifically developed for forensic toxicology and for testing of the long-term alcohol marker PEF. And just before Christmas, we released the 2 × 50 microliter card of Capitainer B, sampling a larger sample volume. And this was also the card you saw the animated sampling video from. And this to enable more analytes to be analyzed with home sampling by a larger sample volume. We will also, in a couple of months, release the first cell-free blood product, filtering out all the blood cells, creating a serum plasma-like sample type. And we also have a urine product under development. Already now we know that the Capitainer B cards works very well with applying urine to the card as well. And there is a publication just now under review for microalbumin on this card. And just talking a bit about the cell-free product that is not on the market, we are calling it at the release Capitainer cell-free blood. Cell-free blood is often used in the analysis of circulating free DNA or RNA, and it's a combination term for both serum and plasma. In this card, we will have a membrane filtering mechanism, so you can at home apply the blood to the inlet. It will go through a plasma filtering membrane. After that, again, as in the other cards, a metering channel, microcapillary channel with a meter and exact volume of filtered serum and plasma, and deposit that to a dried blood spot paper disc. And you use the full metered volume for your analysis. So working with Capitainer in the laboratory, when it comes back to the laboratory, what you do is that you remove the protective tabs on the backside of the card. These are there to protect the sample and also make sure that nothing has happened during the transportation. The sample discs, you don't need, as with standard dried blood spots, there is no need to punch anything out from it because the sample discs are already pre-punched. So in a manual setting, you can just remove the sample discs with a tweezer. They are 6 mm in diameter, which makes them compatible with most standard 96 well plates and also tubes. In the 10 microliter version of the card, you can always also access the excess blood, which is minimum 8 microliter after sampling. And this can be used for non-quantitative analysis. And thereafter, the sample is eluted from the disc in a suitable buffer for your downstream analysis. We know most laboratories working with larger sample volumes will not do manual sample preparation. Therefore, we have, together with a collaborating partner, provide an industrial robot solution provider, developed a semi-automated pre-analytical handler. So this one is very similar in terms of size and in the time you can process your samples to what is standing on all newborn screening laboratories around the world. By using this system, you can scan the barcodes and get full chain of custody and traceability of your samples. You can use it with different plate types, higher plates, lower plates, and also with spin filter plates where you have a cleanup step included in the plate. It also provides an optical disc detection to ensure that your sample ended up in the right well, to further increase the trust in the workflow. And thereafter, you can always export all the sample IDs and the sample spreadsheet to the downstream instrument, for example, an ELISA robot, as an input file for the workflow. We have actually developed for the first customer a fully automated solution for the cards where you can stack up to 200 cards, and it will automatically do all the work with scanning the barcodes, checking sample quality, and transfer the sample discs to the different plates in a fully automatic solution. We will also announce this to the market beginning of this year. Looking at the Capitainer device and what it can be used for, as I mentioned, it's really based on dried blood spots, which means that all the data that is available in the literature from dried blood spots since the 1960s can be applied also to the Capitainer cards, but with the addition that Capitainer makes the sampling much more accurate and also much more user-friendly and with sample protection. We know of customers of ours today working with Capitainer with different analytical techniques within mass spectrometry, and that is, for example, within PKU, but also other small molecules. We have projects ongoing with atomic absorptions. This one is for lithium. We have a lot of work being done with ELISA techniques and also different immunochemistry analyzers in clinical laboratories. There are publications using Capitainer with different multiplex technologies like Luminex or the Olink technology. There's work ongoing with the Quanterix and also with a small instrument provider that I put up here, which is Atana. They are mainly towards the pharma industry. They are active, but they are the first one who have actually put Capitainer into their CE-IVD labeled kit for quantitative COVID-19 antibody diagnostics. So looking, for example, what it could look like if you're working with an ELISA setup and to adapt Capitainer into that. Well, first of all, the sample is detached from the sample card. For the 10 microliter version, the different labs that are using it usually apply 100 or 150 microliter of elution buffer to the sample discs. Usually for proteins, PBS twin buffer is what most people use. Put it at gentle agitation for approximately 30 minutes to an hour. The elevate is transferred to a new 96-well plate, or if you use a plate with a spin filter, you just spin down and get the elevate automatically into a new plate. And then this elevate is used in the ELISA according to the established protocol, where the input volume is adjusted for the dilution factor you get after the dilution of the sample. As I mentioned in the beginning of the presentation, our device was the Capitainer B device was launched to the market in February 2020, just 1 month before the pandemic hit the world. A lot of different studies were initiated and a lot of work, but during the pandemic also many laboratories, both on the clinical side and on the research side, focused a lot on COVID-19 diagnostics, and therefore Capitainer was also involved in a lot of different COVID projects. Although COVID is not what we are going to work with in the long run, it has created some extremely good reference data and proven the concept of using Capitainer both in clinical diagnostics and in large population screenings. So one of the Swedish healthcare regions in the northern part of the country, sparsely populated, decided to implement Capitainer for the COVID-19 antibody diagnostics. In the beginning of the pandemic, anyone could order a free antibody test as well as a PCR test. And in order not to burden the healthcare system with phlebotomy services, they decided to implement with self-sampling in Capitainer. Thereafter, the Public Health Agency of Sweden has chosen Capitainer for home-based sampling in all the seroprevalence studies that has been performed in the country. So from this, we have received a fantastic usability testing of the device in home sampling settings. So for these different COVID studies, over 10,000 first-time users have used Capitainer and sampled themselves at home with only a written instruction. And in the laboratory, they see that over 95% of the samples meet the quality criteria for analysis and are correctly sampled when they come back to the laboratory. And these are, of course, numbers that we are extremely pleased to see, and it really shows that home sampling of blood with a quantitative dried blood spot device actually works. Currently, there is also a large post-COVID vaccination study ongoing in 5 different Swedish healthcare regions in elder care. And here again, they see very good results with this type of sampling, where it's actually can be quite hard to do a phlebotomy on some of these patients, especially patients with dementia, that sometimes can be hard to sample and also aggressive at sampling. So they are extremely pleased with doing the dried blood spot sampling with Capitainer. I also mentioned a bit of PKU and newborn screening. That is kind of the base for dried blood spot sampling. And we are a number of devices out on the market doing quantitative or volumetric dried sampling, microsampling. And in a recent publication that came out this second half of last year in the UK, they had looked into the monitoring of PKU positive patients with Capitainer and other microsampling devices. PKU is what is detected at birth, and then usually with standard dried blood spots, if you lack this enzyme, you have extremely high levels of phenylalanine or very low levels. So a semi-quantitative device actually works pretty well. But going into monitoring of these patients, then the accuracy of the analysis gets really important. And they have seen that the variability in standard dried blood spots is fairly high. It's approximately 20% CV, and that affects actually the accuracy and the way they can monitor patients and decide on drug administrations. So in a publication, they looked at volumetric dried blood spot devices and saw that using Capitainer improves the analytical accuracy. It further improves the analytical precision. It provides hematocrit independent volume compared to standard DBS cards. And it enables a more adequate dietary regimen and recommendations for the patients by moving to Capitainer. And they also saw that it provides better data to evaluate the responsiveness of drug treatments and decide who is going to be put on a certain drug or not. They compared us to some different solutions. I don't know if you have been looking into microsampling devices before. You might be familiar with some of the other devices that was evaluated here. So they looked at liquid blood, comparing that to standard dried blood spots, and here it is not done. It's just a replicate of the same sample, so you don't have the effect of hematocrit variance, etc. They also tried the dried blood spots where they simulated what's being done on Capitainer, but they applied with a pipette an exact amount of blood, the dried blood spots card, and used the full amount. They also evaluated another microsampling device called the Hemaxis device, and they evaluated a device from Neoterix called the Micro device and the Capitainer device. And what they clearly could see from this data is that the Capitainer microfluidic QDBS solution clearly provides the best precision of all devices possible on the market. It's actually also quite hard to pipette whole blood. So the accuracy actually with the dry blood spot solution for Capitainer is much better than pipetted whole blood. And they saw this over different concentrations and for both phenylalanine and tyrosine. And thanks to this, Capitainer is right now being implemented in the UK for the monitoring of all PKU positive patients nationwide. Another application where Capitainer has been evaluated, and this publication came out just a couple of weeks ago. It's looking into a multiplex panel of immunosuppressive drugs to monitor that. And then again, they compared Capitainer with the QDBS technology towards standard plasma concentrations. They also looked at creatinine in parallel to this as a biomarker of kidney function in transplanted patients. And this paper, which is available online, again clearly could show that you have an extremely good correlation with samples taken by QDBS technology and dried blood spots or the standard liquid blood solution. We have currently there's a lot of development ongoing. The commercially available implemented solutions with Capitainer is within COVID antibodies, and it's Unilabs here in Sweden have implemented Capitainer for alcohol monitoring with the marker PEF. But we also have a number of different groups now ready to start implementing Capitainer. We have the PKU monitoring, that's number 2 that I've already talked about, being implemented in the UK. We have another group looking into kidney transplanted patients, looking into immunosuppressants, creatinine and hemoglobin. And again, here they see that home sampling with Capitainer can be on par with what you do today in the standard routine. And this will enable these patients to monitor themselves at home. We have a laboratory that has come very far with the thyroid hormone monitoring for T3, T4, and TSH. A number of groups looking into diabetes patients, monitoring of HbA1c. Work ongoing on CRP, lithium as a drug for bipolar disorder. Microalbumin in urine, a publication is coming out on that, where they actually use the Capitainer blood card, but apply blood to it. We have a project for lead and other heavy metal screenings, and here especially the aspect of the protection of the sample is extremely important, because it has seen that if you do this type of screening in environments where you might be exposed to heavy metals, it's usually a lot of heavy metals also in the environment around. So it's a big problem with a standard dried blood spot approach with contamination from the environment, which they do not see in the Capitainer cards. On the research side, I just mention a few areas. We have quite a lot of work ongoing within the neuroscience field, where they have looked into neurodegenerative biomarkers. We have within immunology and multiplex immunology with the Luminex platform, and also a lot of projects ongoing within the area of alcohol and drugs of abuse testing and many more. Here is just some references I'm happy to provide you with. We have the sampling videos online. We also have a short guideline on how to use the devices in the laboratory. The accuracy and precision publication in PKU monitoring from the UK. And we also have from the Public Health Agency of Sweden a publication on the successful sampling rates using Capitainer in a home sampling setting. So to end this and to give some time for questions, which I'm sure you have, the idea of we really build the devices to be used for clinical diagnostics, and our vision is that we believe in equal access to healthcare diagnostics for everyone, everywhere. But as I mentioned, we are not limited to diagnostics. We see that Capitainer also has a big role to play in larger screenings, in research, and in drug development. So with that, I would like to thank you for getting the opportunity to present the Capitainer solution. I've tried to limit the amount of material to present here to allow times for questions, and please do bring on the questions you might have.
Hi, Michael.
Nice.
I have a question. Thanks for the presentation, and I would like to ask you if is it possible collect in the pet blood in the veterinary field?
If you can collect veterinary blood on the devices, was that your question?
Sorry, the question is?
Sorry, can you repeat your question?
I think is it possible collect pet blood in veterinary field, in the clinically field application? So
yes, we have a few groups looking into also the veterinary field. What we have seen is that what might be a bit hard is actually with the 10 microliter version to apply from a pet or some type of animal directly to the inlet of the device. But now with the release of the 50 microliter card that we launched just before Christmas, we see that the side filling mechanism will likely make it much easier to sample from in the veterinary field than it does with the 10 microliter card. However, although if you are looking into perhaps laboratory animal field, for example, with the rats that you might sample from the tail, etc., then I think the 10 microliter card will work extremely well. We also have one laboratory that was doing a lot of alcohol and the drugs of abuse testing from patients that might be a bit hard to sample. They used a Sarsted Minivet that collects 20 microliters to actually get the blood in, have some time and to collect the blood into that before applying those microliters directly to the Capitainer card for measurement, drying and shipment, etc. So that could also be a solution for veterinary use.
Thank you. Thank you very much.
Hi, Michael.
Hi.
Here is Carlo. I have a question. Have you ever tested your device for samples other than bloods, but commonly used like urine or saliva, body fluid?
We have first of all, we are device manufacturers. So we don't have our own lab. We have a development lab and we have a lab for quality testing, etc. But all other testing is done in collaboration with other laboratories where we have an active collaboration. We have tested urine with the 10 microliter card. That is done. That publication is under review at the moment, and they have done that for microalbumin, which is the albumin creatinine ratio, and so that has been shown to work fine. The idea is only to measure a very accurate amount of fluid. So it likely works also for other body fluids. What can be a challenge with saliva is that it might be rather viscous. And since we measure the volume in a microcapillary, I think there is a risk that will clog. If it's not not a pure sample, it might contain particles, etc., that might cause a problem in this microcapillary measurement.
But maybe for research and development, if somebody wants to test it or to try it, they can go ahead.
Yes, of course.
And Michael, this device collects only whole blood sample, or is there the possibility to collect only plasma, for example?
Yeah, with the new Capitainer CF device that I mentioned, then that will have a membrane filtering function in it. It's planned to be released in March. We are already doing together with our collaborators some quite a lot of testing of the device. So that is possible. It's also a possibility that we know for some analytes and in some laboratories, they are actually just interested in the ease of shipping and the stability of the analytes in the dried format. So we have one group at the moment working within neurodegeneration, looking into Alzheimer's. They are only interested in the quantitative aspect and to be able to dry and ship the samples, not really the filtering mechanism. So there it is an option also that they want to do a traditional blood draw centrifuge plasma, but they want to apply it on a device that you can easily ship to a laboratory that is far away from the site where you take the samples. And that we know works, of course, as well on the blood device if you applied a serum or plasma sample onto the device. But then it's not really a home sampling setting to create a serum or plasma sample. Then you need a new device that will be launched in a couple of months.
Perfect.
I can also take, I took a device here just so you can see it. I can show this is what it looks like. So it's extremely slim and thin. And the idea of that is, of course, also to add to the ease of shipping. We have been very careful to take it with the sustainability aspect of the device. So it's actually mostly is based out of paper. Over 80% of the device is paper based. Less than 20% is plastics to give it a good sustainability profile as well.
Good. Are there any other questions? In conclusion, for several applications, what are the main advantages compared to traditional dried blood spots collection system?
Yep.
In addition, of course, to the fact that there are quantitative.
Yeah, I mean, one main advantage is, of course, the quantitative aspects, and looking into many analytes, you need to have a good quantitation. But it's also, I think it's also the contamination aspect we see is very important. I had a meeting not too far ago with a big metabolomics group in Australia who described that they have tried to do metabolomic studies with standard dried blood spots, and it happened now and then that they started to get that they got some really weird results from these samples sent back from people's homes. And when questioning that, it turned out that they had actually done the sampling at home on their kitchen table and put the cards on the kitchen table, and most likely there were some contamination from their breakfast, some spilled milk or something on the table that actually got onto the card at the sampling point and then was transferred into the analysis. So I think the sample protection is really important, but also the workflow in the lab. This is something we have heard from many laboratories doing, especially research studies with the card, that when the sample comes into the lab, you don't have to judge if it's okay or not. It's easy. The samples always look consistent. They are the same. You know that if it's blood in the spot, you know it's a perfect sample. So it's never any question if this might be a good or bad sample, and you don't need to, as with traditional dried blood spot cards, do this decision where in this blood spot are you going to do the sub-punch, because that will also affect. And usually, even though you might have quite some blood in a traditional dried blood spot card, it might not be so many places where you can punch out a good quality sub-punch from that dried blood spot card. So the fact that you have it pre-punched, it's designed to fit in a standard 96 well micro titer plate, easy to use, you know the sample quality is good, also gives trust to the sample and the accuracy when it comes into the lab.
Okay.
And also looking in one of the first seroprevalence studies, which was done in Sweden during the pandemic with a research group, that was done with the Capitainer device far before any public initiative was done. And they also had the possibility then to do it with a Luminex panel and see that it is compatible to do a multiplex serology approach on it, so they could look at all different serotypes, etc. And with the stability of the device, it was easy for them to bring all the samples in for a couple of weeks and analyze all at the same time. Because compared to the liquid form of blood, in the dried form, there is almost no enzyme activity which can break down the analytes in the device, which means that the stability in room temperature is kept very well. So that's another feature for storing the samples. You usually don't need to store them in -80 degrees. It will of course be a bit analyte dependent, the stability also in dried formats, but looking at most publications, you never need anything else than -20. In dried formats, it's very little benefit going from -20 to -80. And also many analytes are stable in weeks up to months in room temperature.
Yeah. Thank you. Any other question?
Thank you.
Okay. If there are no other questions, we can say goodbye.
Okay, thank you.
Thank you, Mikael, for
Thank you, Mikael.
this clear presentation. Thank you, everything, for participating. I put in the chat box my contact details so that you can use for any further information.
Thank you very much.
Thank you for getting the opportunity to present, and please get in touch with Ilaria if you have any additional questions, and we are together happy to support you.
Perfect. Thank you.
Thank you very much.
Bye.
Bye-bye.
Bye.
Thank you, bye.